Good morning. My name is Anja Weber, and I'm presenting today on the topic of soil decontamination techniques. This is quite a scientific topic, and therefore I will use it to demonstrate to you how illustrations and animations can help us to break down content that is otherwise quite complicated to communicate. And my goal for today is that at the end of this presentation, you will have understood why the topic of decontamination is important to us, and that you will hopefully, at the end, will be able to identify some of the major techniques used to decontaminate soil. To do that, I will, as I said, use a PowerPoint presentation with illustrations, especially when we're looking at alternative decontamination strategies, but we will also tackle one or two questions regarding contamination. And for that, I would like to ask you to be forthcoming with your contributions, and for the sake of our only limited time of 20 minutes, to keep your answers as brief as possible. Thank you. At the end of the presentation, I will test your knowledge to see if, or to evaluate if this presentation method was successful. So please pay close attention. If we want to understand decontamination, I think we should start with a definition of what contamination is first. Soil contamination is defined as the presence of toxic substances in soil in concentrations that exceed regulatory levels and are therefore a risk to human health, as well as the environment or ecosystem. When you look at this picture, which sources of contamination can you identify? Can I please have 4 people state 1 each? Okay, well done. Let us summarize together. We are looking at pesticides as the most common contaminant. These can be caused, as you said, by pesticide spillage, but not to forget also from washing the equipment or the protective gear. And it, of course, can come from cattle dips, where we also might have spillage or contaminated water. Pesticides are dangerous because they affect the nervous system. They can cause dermatitis as well as cancer. The second most common contamination stems from nitrate and phosphate, which is caused by, as you said, fertilizer overuse or spillage, but as well from livestock, uncovered manure piles, defective sewers, and contaminated runoff. When entering open waters, nitrates and phosphates cause eutrophication, which, as you probably recall, causes excessive growth of algae and depletion of oxygen in the water. The decaying algae, however, can also produce toxic bacteria. Ingestion of nitrate can cause a condition called blue baby syndrome, which is the result of lack of oxygen in the blood. And this can, of course, result in serious illness and even death. One of the most dangerous contaminations is that of heavy metals, which come from waste dumps where we might have old batteries, paint tins, and so on. But it can also come from contamination in the larger environment, like from mining is a very dangerous source, or from industry. And then we might have old machinery or vehicle batteries sitting in the yard, or there might be spillage in the workshop from oils, solvents, and so on. If we look at one particular example, lead, that is very, very dangerous, toxic when taken in even in very small doses, especially for young children or if taken over longer periods. It causes neurological damage and can even be fatal. Dioxin, the term dioxin is used for a group of chlorinated compounds, and they are the major concern from burnt waste. They can also cause cancer and cause great damage to the fetus during pregnancy. In order to understand why soil contaminants can become dangerous to humans, I think we need to look at the way contaminants take from the soil into our bodies. Contaminants can be carried with runoff water and run into open water sources. They can be dispersed by wind erosion, and of course they can leach from the soil into lower lying areas or into underground water sources. Then contaminants can also be taken up again with the water if we drink it, or if we use it in the house for our bath or for cleaning. Contaminants can also be taken up by plants like food crops and by animals, either directly from the soil or with contaminated feed. And then in the last step, eventually, contaminants are taken up by humans once they eat the contaminated food. The best way to avoid hazards from contamination is, of course, to avoid contamination altogether. But to do so, we need adequate construction, safe procedures, and regulation and controls. But if contamination has already happened, the standard technique of decontamination is excavation. That means the contaminated soil has to be safely dug up, removed, and destroyed by incineration or disposed in a safe location. The problem with this strategy is that it's first costly, it destroys the area, and we still have the problem that we do not know what to do with the contaminated soil. Let's do a quick reality check. Are avoidance and excavations solutions for ACP countries? Yes or no? Give me a quick answer. Well, I think so too. And this is the problem. But this is also where the alternative strategies come in. And this is what I want to take a look at in more depth now. Alternative strategies, these are techniques that make use of natural processes that we can harness to decontaminate soil. The first one is called bioremediation. Bio is meaning life. So this is a technique that makes use of living organisms. And remediation comes from the word remedy, meaning solution. So this is a strategy based on increasing microorganism activity, not to remove the contaminants, but to quickly degrade them to safe levels. The second strategy that we will be looking at is called phytoremediation. The word phyto meaning plant. So this is a remedy that is based on the use of plants and their ability to take up and accumulate contaminants in their soils. This is a strategy that can help us to quickly reduce the danger from contamination, especially by inhibiting its spread into the larger area. As I said, bioremediation is a method that makes use of microorganisms to break down contaminants. This is basically the same technique as it used in composting. And this method is particularly useful to break down nitrate and phosphate, but can also be used to remedy contamination from some pesticides. To employ this method, we have to engage in activities that increase microbial activity, like increase organic matter content in the soil, raise soil moisture levels, and increase aeration, for example, through movement of soil. Bioaugmentation is a special case of bioremediation where microbial cultures are added to the soil to speed up the process. The difficulty is that these cultures can be expensive and they're not always available. The other strategy, phytoremediation, employs plants that have either the capability to accumulate large amounts of toxic substances without suffering damage themselves, or that can break down such substances. And as I mentioned before, This can also just be a simple plant cover used to hold contaminants in place to avoid the spread over a larger area. These strategies have the obvious advantage that they can be used in situ, which means on the site, so no excavation is needed. They might therefore be cheaper and less destructive. On the other hand, these strategies, of course, depend on natural processes, and they are therefore very difficult to steer or monitor, and they take their time of the natural course. It's a natural process. So this can be a very long-term solution, and in the worst case scenario might mean that an area is taken out of production for 5 years or more. I want to illustrate some of the phytoremediation techniques in more detail. Let's firstly look at phytoextraction. So this means the uptake of contaminants from the soil through plants. The next technique we will be looking at is somewhat similar. It is called phytosequestration. So in this case, it means forced uptake of contaminants by plants. Then we have phytostabilization and phytodegradation, which is the breakdown of contaminants through plants. And the last one that we'll be looking at is called rhizodegradation, which is closely related to phytodegradation, but in this case, the breakdown happens through very particular activities in the root zone. In phytoextraction, contaminants like heavy metals, for example, are first dissolved in the root zone. And then they are absorbed into the root together with the water through the normal transpiration channels. Inside the plant, they are formed into chelates to render them harmless. And then they are moved up into the shoots where they are stored. One example of a plant that can be used for this is vetiver grass, but any other plant that quickly produces large masses of organic matter, like seen here in the picture, are effective in this way. The difference between phytoextraction and phytosequestration is that the contaminant, like metal compounds, is not first dissolved, but taken up by the root directly. That is possible through protein transporters in the root wall. They actively take the contaminant in and then store it in the cell walls. An example for such a plant is the Cisbania pea, but many aquatic or semi-aquatic plants are also very effective in this. Phytostabilization is the process in which contaminants are not necessarily taken up, but held in place by a soil cover that prevents erosion. They can also be held in the root zone, or they can be attached to soil aggregates. This technique is used to prevent immediate danger from very toxic substances from spreading the contaminant to the larger area. And it can, of course, be used together with any other bioremediation techniques to dissolve the substance long term. An example for this is the Peruvian pepper tree, but generally all fast-growing trees or ferns can be used for this. Now let's look at phytodegradation. In this case, the plant secretes enzymes from its roots to actively break down the contaminant like petroleum products or herbicides. In some cases, this can also take place inside the plant. The disadvantage of this technique is that it is slow and only works in the immediate area around the root, so not in lower laying levels. A plant that can be used for this is pigeon pea. In rhizodegradation, on the other hand, instead of enzymes, microorganisms that live in close association with certain plants break down the contaminant. Many kinds of brassicas, like Indian mustard, are successful in this technique. Of course, there are other strategies to decontaminate soil, like soil washing, thermal treatments, and so on. But to be honest, none of these are feasible under normal ACP circumstances. I'm now really curious if you can recall what you have learned Let's give it a try with this little quiz. Please unmute yourself and try to complete the following sentences. Well done. Let's give it another try. Great job. Thank you very much. So, do you think using illustrations to present this topic was helpful? I'm really curious to hear from you. And if you have any further questions, please do not hesitate to contact me under this address. Thank you for your attention. I hope to talk to you soon. Bye-bye.